The reagents and supplies which are required for maxi prep are going to include even those reagents which we need for the microorganism culture. For this purpose, we need ampicillin, which will be used in the bacterial culture. Then we are going to need a series of buffers. Like buffer P1 is referred as resuspension buffer. This will be used to resuspend the bacterial cells in this buffer before we can proceed for lysis. The second buffer is P2 buffer. This nomenclature is not standard. You can have your own nomenclature for these buffers. Buffer P2 is a lysis buffer which is going to be used for lysis of the bacterial cell. This contains the detergent and alkali in it. It can be stored at room temperature. Buffer P3 is neutralization buffer, which will be used after lysis to neutralize the alkaline solution or the effect of the alkaline solutions. Now we have to use those buffers for the columns. The QBT buffer is referred as column equilibration buffer. It will be used to equilibrate the column. QC buffer, also referred as wash buffer or quality control buffer. QF or the elution buffer will be used to elute the DNA sample. All these buffers have variable concentrations, different storage conditions. So being a researcher, you need to follow that the storage conditions are to be maintained and the composition and the pH should be observed. Beside this, supplies which we need during this maxi prep, we need centrifuge bottles, the larger one, 500 milliliter for harvesting the bacterial samples, tube racks, centrifuge tube racks, then centrifuge tubes, sterile. Again 50 milliliter centrifuge tubes which are going to be used separately to collect the eluted sample. We need ethanol 70% which will be used as cold. Then ice, isopropanol, the LB broth culture medium, pipettes, TE buffer, and the kits required for maxi prep, which will include those resin columns which will be used for elution and purification of the DNA samples.